Okay, can somebody confirm you can hear me?
Yes.
Thank you. Let's go ahead and shut that down. There we go. I think we're on chapter two. About there. Is that where we are? Yep. Okay, before we begin, are there any questions? Let me share my screen here. All right, you should be seeing my screen now. Hopefully you guys had a happy Easter. Uh, Today is April 22nd, and we are supposed to be finishing chapter two today and hopefully starting chapter three. I don't know if we'll make it, but hopefully we'll finish chapter two. And there will be a lecture today, lab four, in the lab. There will be a quiz one, covering chapter one, chapter 10, the start of chapter two. So anything we've covered in chapter two up until anything today. We're not going to talk about the quiz, will not cover anything we cover today. And then lab zero, one, and two will be on quiz one. This will be your first real quiz. Please make sure you have a stable and long internet or strong internet connection before you take it. If you do not have a strong internet connection or a fast internet connection, you should probably go to Clark College and take the quiz there. Either hook onto their network with your own computer or they have computers at Clark College. I think they're in the library, but I'm not sure where they are for using Respondus Lockdown Browser. You should let me know in case I need to flip a switch on the Lockdown Browser if you're using the Clark College computers, and then it will work if I flick the switch. I think if you use the Clark College computers, you will be proctored, and you may not use, oh, what is it, Respondus Monitor, but you will be proctored by someone from the college. But like I said, I need to flip a switch on the exam. So if you use a computer at Clark College, just let me know. Also, if you want to take the exam Wednesday night, please let me know and I will print it out for you. And then I'll go into Clark College. If I don't hear from anyone that they want to take the exam in person, I will not print it out and I will not go to Clark College. So if I don't hear anything, I'm going to assume that everyone's going to take the exam over the internet. Okay? Which is fine as long as I can see the environment around your computer. If you take the exam over the internet and I do not see the environment around your computer, then I will watch your eye movement during the entire exam. And as long as you keep your eyes on the monitor of your computer, you will be fine. However, if you take your eyes off the computer and it looks like you're reading something, meaning your eyes are not on the computer monitor, I'm going to assume you're cheating and I will mark you accordingly, meaning you'll get a zero on that exam. Now, if you show me your complete computer environment and you take your eyes off, especially if your eyes are just taken off the computer monitor for a brief moment of time, that will be fine. But if you do not show me your complete environment around the computer and you take your eyes off the monitor, I'm going to assume you're cheating. Any questions about any of that? All right. Up, it says my internet connection is unstable, so if you don't hear me or the internet connection breaks up, just let me know and I will repeat what I just said.
I think it was towards the ending, but I'm not sure for everyone else, but I didn't catch that ending portion.
Which,
Yeah, I
Either.
didn't
which
I
catch
was
it
didn't
what?
either.
see.
Which was what? The ending portion. Where did I leave off?
If we look away after we show you our environment, what happens?
If you do show me your complete environment and you look away for a brief amount of time, it's fine. Okay, because you've showed me your environment, there won't be anything around for you to cheat with. But if you do look away for a long period of time and it's obvious you're reading something, I will know you're cheating. Okay. But if you cannot show me your complete computer environment, meaning the environment around your computer, and you look away from the monitor, I will assume you're cheating. And that's why you're not showing me your complete environment. Okay? Another thing, a few of you, I gave a warning that you're reading the exam question out loud. If I can hear you, so can an AI. And so don't read the questions out loud. You may mouth the question where your lips move, but you're silent. If I can't hear you, an AI can't hear you. And in AI, they are set up now where, at least some of them, you can get it so they will always respond to you and you can get their response in text. And so you cannot read the questions out loud. And I'm sorry about that, but AI have become very complex now, and you can get any answer from an AI. So you cannot read the questions out loud. And there's only a few of you who are reading it out loud, but there were a few. All right, any questions about the exam?
I have a question about the homework.
Okay.
So for me, you said that I could repeat one of them and send it in for grading again. I just want to confirm, is that part of a grade, or if I correct it and send it back, it would be 100, depending on what I get?
If it was week two, all of the labs in week two can be corrected for 100%. You have until this Saturday to turn in the correction. Now, for week one, you only got it graded one time, okay, for the lab. But for week two, all of the labs may be graded two times. I have not looked to see how the class voted, and how the class votes will depend how weeks three through, I don't know, week nine or 10, meaning week up until lab nine, whatever week that is. Uh, it will go as the class votes. I have not looked to see how the class voted, so I don't know the answer there. All right, any questions about that?
I
For
had a
the
quick, oh,
lecture worksheets, you only are turning in a question or two. And so if you don't know the answer, please look it up because it will be only graded one time. All right, somebody had a question?
yeah. You said that my webcam went out a few times and then my internet, but I have the high speed internet and then I sit right next to my modem. So I was wondering if there's anything I can do to fix that for tomorrow in case it does the same thing.
I don't know. It broke briefly if you're the person I'm thinking of, so that the video recording if you're taking the exam was fine. It was recorded, but there were a whole bunch of messages saying that the camcorder shut down, the camcorder had to restart, and there was a few times where I got a message, I don't remember how many, two or three times, where it said your internet connection broke. I don't know if you got the warning or not, but I got the warning. I don't know what the problem is, but that was a short exam where I got all those messages, and you don't want that happening on a longer exam. This exam will be close to a half an hour.
Yeah, I didn't get any messages, but it was briefly showing what I look like on the screen in between like multiple, I want to say like the first 8 questions.
Yeah,
And it was really distracting because it was like flipping from
back
that
and forth.
screen to like
What
the
was
question.
what was happening was the camcording stopped, and I imagine it showed you your picture at that point, and it was
Is there a way
probably.
that I could fix that? Because I
I
tried
don't
looking into
know.
it and asked
I don't
the
know. Like I said, it was your internet connection that was the problem. I don't know what the problem is. So I'm not
hmm,
sure what was going on. Okay. Um,
uh okay.
like I said, I suggested you could take your computer to Clark College. They have a strong internet connection. If you normally have a strong internet connection, maybe your internet connection just during that one time was, I don't know, interrupted or something.
It's a desktop and I was checking the speeds after when you said that, and maybe I should
Well,
take a picture
afterwards
of
would be
them.
a long time afterwards because I obviously was looking through people's environment during that quiz after the quiz. So it wasn't right after you took the quiz.
Okay. I just didn't want it to like affect my grade then.
Well, as long as the quiz you can take it, it won't affect your grade. Other than you might be confused what's going on, but you know you have a weak internet connection when you see your picture coming up. And I'm surprised you never got a message saying what the problem was, because I've got it in there that if there's a problem, that the um, lockdown browser is supposed to notify you during the quiz. And I'm surprised you
Mm.
never got a notification because I got, it was something like 12 different notifications.
Yeah, that's what you said. I didn't see anything except for what it like flashed my face really quick and then went back to the question.
Okay, not sure what's going on. All right, let's continue with the exam. We need to talk about lipids. Lipids consist of carbon, hydrogen, and oxygen. There is not much oxygen in most lipids, but there is some. And there's always a lot of carbon and hydrogen in everything which is a um, what is it? Organic molecule, and lipids are an organic chemical molecule. So there's always a lot of carbon and hydrogen. Lipids are mostly nonpolar and insoluble in water, meaning lipids are hydrophobic. There is one exception we will talk about. Lipids are structural and energy storage, play energy storage roles in cells and living organisms. Lipids are primary components of the cell membrane, meaning your cell membrane, the main molecule, is actually a lipid. You can break lipids down into 3 types: simple lipids, complex lipids, and steroids. Any question about any of that? All right. The first simple lipid we're going to talk about are fatty acids. They are a major components of some lipids, most lipids. They consist of chains of carbons. They do have a carboxyl group, COOH, at one end. Let me see if I can blow this up. Sorry, I've got a frog in my throat. There we go. So there's the carboxyl group at one end of the fatty acid chain. They mostly lipids are carbon hydrogen chains, where carbon's the main molecule holding the molecule together. We have 2 types of fatty acids: a saturated fatty acid, which has a single carbon bond holding all the carbons together, a single covalent chemical bond holding all the carbons together. So that's a saturated fatty acid. And an unsaturated fatty acid, which has one or more double carbon bonds holding the carbons together, meaning there would be 2 carbons, like right there, 2 chemical covalent bonds holding the carbons together. wherever you have a saturated fatty acid, or meaning the double carbon bond, you'll have a kink in the chain, which is shown right here. Let me see if I can blow that up. Right there, we have a double carbon bond holding the two carbons together. And as you can see, there's a kink in the chain right there. You only need one double carbon bond holding the two carbons together in the entire molecule to call this an unsaturated molecule. And this is the fatty acid right there. So that's the unsaturated fatty acid. This is a lipid, meaning we have three unsaturated fatty acids, 1, 2, 3, hooked together with the glycerol molecule right there, making a fat or a triglyceride. Both a fat and a triglyceride are the same thing. It's different words for the same thing. Excuse me here. That didn't help. Try the water. You only need one double carbon bond in the molecule, and this is an unsaturated fat or unsaturated fatty acid. Let me shrink that down. Oh, wrong way. Triglycerides, by the way, are the most common fat in your body. You can just look at your belly and you can see some triglycerides there. A triglyceride is three fatty acids hooked together with a glycerol molecule, making the triglyceride, also called a fat. Fats are the body's richest energy source, stored as adipose in animals, meaning fat in an animal. What happens is that a fatty acid just has a OH group come off and an H group come off the glycerol, and then that carbon links directly to that oxygen right there. I mentioned that a organic chemical molecule has mostly carbon bonds linking the molecule together. And if you look here, all of these links are carbon links. There's an exception, and oxygen's holding the molecule together. But once again, we have carbon links. And right there, we have another exception, oxygen holding the molecule together. And then there, we have only carbon links holding the molecules together, meaning carbon is the main molecule holding the backbone of the molecule together. Any questions about any of this? Now, fats are the body's richest energy source. So if you ever were stranded in some place like Point Barrow, Alaska, and you had a bunch of food in your car, what you should carry, let's say you got to walk 20 miles in the snow and cold weather to get to Point Barrow, Alaska, and someplace safe. And the thing to carry would be fats because it will give you the most energy for the amount of weight that you'd be carrying. So you get more energy from fats than any other food source, like protein or carbohydrates. It might not be the healthiest thing for you to eat, but you get the most energy. And like I said, if you needed energy, which you would need energy in Point Barrow, Alaska in the wintertime to keep you warm. Any question about any of that? All right. So that's simple lipid, triglyceride, and fats. Another and fatty acids. Another simple lipid is the phospholipid, which is different from all other lipids, and that is a phospholipid has a polar end. That's the phosphate group and then a nonpolar end, the fatty acid group. And so the polar group is actually a molecule which can dissolve in water. So phospholipids can dissolve in water. And then the fatty acid end of the phospholipid can dissolve in lipids. So this molecule is biphasic. It can dissolve in both water and in oil. There are not too many molecules which are biphasic. You actually know of, well, a few, like soaps and detergents are biphasic, and that's why we use soaps and detergent to clean our clothes, and that is the water will dissolve the polar molecule out of our clothes, and then the soap and detergent will dissolve the oil or what is it, the lipids out of our clothes. And that's why we add detergents to our laundry, and that's because it'll clean our clothes better than water alone. Or if you were to use, I don't know, like a lipid to clean your clothes, the detergent will clean the clothes better than oil or water alone. Any question about any of that? So phospholipids are unusual, and they are the one lipid which is not nonpolar. It can dissolve in water, and so we call it, at least part of the molecule, polar. Let me blow this up again. There's the phosphate group on the phospholipid. The R just stands for any organic molecule here, so that can be another, I don't know, complex carbohydrate or complex molecule right there. Anyways, the phosphate group is polar. In fact, it's charged, as you can see. The glycerol molecule is slightly polar too. So the head of the phospholipid is polar, and then the tails, the fatty acid portion, are non-polar. So there's the phospholipid shape. And when we put a bunch of these in water, they will actually start pairing up like this, where the head will be facing the water here and facing the water there. And then the fatty acid tails, which do not like to be around water, come together. And let's go on further. And then they'll wrap around each other like this or like that. We'll talk about that one there. And there you can see outside the liposome, the heads will be facing the water. The nonpolar tails of the phospholipid are on the inside away from water. And then we'll have the heads on the inside with water inside. So the heads over here on the inside are facing the water inside the liposome. And what does this look like? That should remind you of something. What does that look like? Come on, somebody's got to guess.
The thing on the plane, the jet or I don't know what it's called.
What is your body composed of? What does this look like in your body? Like in your blood, for example. It looks like a cell. A cell is mostly phospholipids in the membrane of the cell. And so we'll have this phospholipid membrane composed of two layers of phospholipids with the heads being outside the cell. Think of the cell in blood. There's mostly water in blood, so the heads are facing the water. And what's inside the cell? Cytoplasm, which is also mostly water. And so on the membrane, the heads of the phospholipids are facing inside the cell, which is mostly water. It doesn't matter well where the cell is, mostly outside the cell is water in the body. And so the outside of the cell will be facing the water. Any questions about that? Let me see if I need to go back a little bit. Oh, yeah, I do. I want to talk about one other thing right here, and that is our membranes, in fact, all membranes, have a little bit more than the phospholipid in the membrane. It is the main molecule in the cell membrane, but you can also find carbohydrates and proteins and lipids, especially lipids in an animal cell membrane. You can find all of those, but the phospholipid is the main molecule in the cell membrane. In animals, you can find cholesterol, shown here, which can be found in the cell membrane. Why do animals put cholesterol in their cell membrane? It's because animals only have a cell membrane. They don't have a cell wall, and the cell wall can help protect the cell membrane. So animals put cholesterol in the cell membrane to give the cell membrane a little bit more strength and support. You only find cholesterol in animals, and it's true cholesterol can be found in more than just the cell membrane, but all cell membranes of animals have cholesterol in it. You will not find any cholesterol in any plant-based food or any plant that you're eating. Okay, any question about any of that? You only find cholesterol in animals. You don't find it in bacteria or archaea either, but you do find it in animal cells, especially in the cell membrane. All right, that's it for the phospholipids, the last of the simple lipids. Remember, the simple lipids include, there's the word I'm trying to get, fatty acids, fats and triglycerides, and any other, like a biglyceride or something like that, which is I guess that's not a fat, a biglyceride. And then phospholipid. Those are the simple lipids. Let's talk now about the complex lipids. There are steroids and sterols. Steroids are four interconnected rigid carbon rings characteristic of the group. Like right here, here we have a 1, 2, 3, 4 rigid carbon ring of a steroid. If there's an OH group attached to the steroid, we call it a sterol. So this has an OH group on it, and so it's given the name sterol. Without the OH, it's called a steroid. Cholesterol is a sterol, and it's an important part of the animal plasma membrane. It's a molecule you can only find in animals, cholesterol. Any question about any of that? All right. Proteins is the next topic we want to talk about. Proteins are essential in cell structure and function. Proteins play a lot of roles in cells and in living organisms. Proteins may be structural proteins found in bone, muscle, and hair. Structural proteins tend to hold things together. Proteins can be enzymes, molecules which speed up chemical reactions. Proteins can be transport proteins that move molecules across a cell membrane or a plasma membrane. Proteins can be regulatory proteins. They regulate the body functions, like a growth hormone can cause a child to grow. Proteins can be responsible for movement. The reason why my arm is moving is because of the proteins in the muscle in my arm. The reason why a bacteria can move is the proteins in the flagella. And the reason why we walk also. So proteins are involved in movement. Proteins can be responsible for some bacterial toxin. A toxin is a substance which is poisonous to us, and some bacterial toxins are proteins. Actually, most of them are. There is at least one which is not, but most are, the toxins are proteins. And then proteins can be antibodies. These are proteins that help defend the body from a microbial invasion. So antibodies are proteins that our body makes to help defend us. As you can see, proteins are playing many different complex roles in cells. Proteins are more diverse than any other of the biological molecules. Why are proteins more diverse than any of the other biological molecule? And that's because proteins consist of monomers called amino acids, and proteins have 20 different amino acids. At least humans have 20 different amino acids, and most living life organisms have 20 different amino acids. Generally, if they have a different amino acid than we have, they drop out an amino acid and replace it with another. Most living organisms have about 20 different amino acids. They don't, especially the microbes, don't have to have 20. They can be similar to 20. The highest would be 23, but you don't need to know that. Just most living organisms have about 20 amino acids. That's more monomers than any of the other biological molecules. Like carbohydrates have, they're made up about a dozen different simple sugars, maybe even a few less, because some of those dozen might be sucrose and lactose, which aren't a monomer, they're a dimer. And but there's about a dozen simple sugars which make up carbohydrates. And then lipids, which I forgot to mention something. Lipids are made up of a little less than a dozen fatty acids and glycerol. The nucleic acids are made of only 4 monomers, 4 nucleotides. Doesn't matter if it's DNA or RNA, they both are made up of 4 nucleotides. So the other biological molecules don't have as many monomers as amino acids. And let me step back to the lipids. I forgot to mention that the simple and complex lipids are made up of glycerol and fatty acids. And then the phospholipids are also made up of glycerol, fatty acids, and phosphate, or the phosphate group. So they are made up of, or the lipids are made up of, or some of the lipids are made up of monomers. Okay? Like the triglyceride is made up of 1 glycerol and 3 fatty acids. The phospholipid is made up of 2 fatty acids, 1 glycerol, and 1 phosphate group. The steroids and the sterols, though, are not made up of any monomers. So the lipids, some of them have monomers, which makes the polymer, and others, they're just one molecule. So they're not a polymer, they're not a monomer. You would call it a long molecule, but it's not a polymer because it doesn't have monomers making it up. And that's the steroid and the sterols. For the proteins, the monomers are called amino acids, and this is the generalized shape of the amino acid. You'll note that the amino acid has a central carbon hooking the molecule together. It always has a hydrogen hooked to that central carbon, and then an R group hooked to the central carbon. That R group can change, and it changes with the amino acid. So depending on which amino acid it is, it'll have a different R group. So the R group just stands for a biological group, And there's one R group there for showing you the amino acid tyrosine. You don't need to know that, but it's a R group there. And then on the amino acid, you always have an amino group, NH2 on one end, and a carboxyl group, COOH, on the other end. You have many amino acids linked together to form the protein, and you can just hook them together like beads on a string to make the protein. Any questions about proteins and amino acids? You should know this generalized amino acid structure. You don't need to know this. Let me show you a few of the different 20 amino acids. You do not need to know these. I'm just showing them for your benefit. The amino acids only differ on the side chain called the R group. The simplest R group is the amino acid glycine, which I can't see it there, but there's the R group, which is a little darker background on the H. The simplest R group is just a hydrogen atom, and that's the amino acid glycine. You can have a methyl group attached as an R group, that amino acid. You can have several methyl groups attached, this amino acid. All of these R groups attached to the amino acid are non-polar, and they're all methyl groups attached in different combinations. You can have ring structures attached, so that's a methyl group attached and then a carbon ring structure. This, and then all the methyl groups, all of the earlier ones, are non-polar R groups. And then you can have OH and NH groups attached to the R group. That OH group and the NH group make this region of the R group polar. So this group is polar. Realize this entire region of the R group is nonpolar. And then that's nonpolar, that, and that is nonpolar, but this is polar and that's polar. Remember, the polar R groups can dissolve in water. The nonpolar R groups cannot dissolve in water. So a protein will always have nonpolar regions. Like this amino acid R group will be nonpolar. And then it will have polar regions. All the amino acids, the amino group, the carboxyl group, will be polar. And then some of the R groups, like this R group here and this R group there, will be polar. With a protein that is floating around in the cytoplasm or floating around in water, like in your blood, the R groups, which are nonpolar, will be tucked inside the protein so that the R groups, which are nonpolar, or the nonpolar region of the molecule, will be tucked inside the protein so that the polar region of the protein will be on the outside of the protein, exposed to the water. On the other hand, if the protein is in a lipid, like a phospholipid membrane, which is nonpolar or mostly nonpolar, it will have the nonpolar regions of the protein tucked on the inside of the protein. And then did I say that right though? It'll have the polar region of the protein tucked on the inside away from the lipid regions of the cell membrane. And then the non-polar regions of the protein will be exposed to the surface of the protein. And then that will allow the protein to dissolve in the non-polar lipids of the membrane. Any question about that? All right, you do not need to memorize the different amino acids. You do need to know that amino acids are linked together by a peptide bond. So we have two amino acids, this one here and that one there. water comes out in the condensation or dehydration synthesis reaction, and OH comes off of one amino acid, H comes off the other to make the water. And then this carbon will link directly to that nitrogen, creating a peptide bond. That's when carbon links to nitrogen. Peptide bond is rare in biological molecules, it's rare in living organisms, and actually it's rare in the universe. We do not see a peptide bond very often where the carbon is linked to a nitrogen, holding the two, I guess, monomers together. But you do find it in a protein, which is a peptide, and that's why it's called the peptide bond. And you just link the different amino acids together in this fashion to make the protein, which is a polypeptide, meaning it's many peptides or many monomers hooked together to make the protein. Any question about any of that? So all of the polypeptides hooked together, meaning if the growing protein has all of the amino acids put together, linked on the polypeptide, we then call that polypeptide a protein. So as the protein is growing, meaning they're adding the amino acids to it, it is not called a protein. It is called the polypeptide. You can also call it a growing protein because that's implying that's not the finished protein, that more amino acids are being added to it. So while the protein is growing, it's called the polypeptide. Poly means many, so you have to have many monomers, and I forget the definition of how many amino acids it is before it's many, but certainly more than two. And then it's a polypeptide all the way up to the last amino acid which is added, and then it's called a protein. Any question about any of that? So the difference between a polypeptide and a protein is mainly a polypeptide is still growing, and once the last amino acid is added, it's then called the protein. Any questions about any of that? All right. Proteins have 4 different levels to the protein structure. There's the primary level, the secondary level, the tertiary level, and the quaternary level. Let me mention that some proteins do not have the quaternary level. Only some proteins have the quaternary level, but proteins have 4 levels to their structure. And let's watch a little video on the 4 levels of protein structure. After
I
this
have to
commercial.
finish this paper. I'm swamped. No one told me college would be so damp.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of 1.
Let me state that right here, the video is a little bit of an oversimplification. There's more than hydrogen bonds determining the shape of the secondary structure, and I'll talk about that more after the video. I'm just stating that the video is a little bit oversimplified right here.
One amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about that video? So let's talk more about the 4 levels of protein structure. The primary structure is just the specific sequence of amino acids in a polypeptide chain. So when the protein is first made, all it is the primary structure where there'll be one amino acid followed by another. And that's the sequence of amino acids in the protein. However, the amino acids can soon begin interacting with each other, and that gives rise to the secondary structure of the protein. The secondary structure of the protein is localized interactions between neighboring amino acids. So it's only the neighboring amino acids or nearby amino acids which cause the secondary structure. Let me blow this up a little bit. Right here, we have a hydrogen bond forming between this amino acid and that amino acid. And that is an interaction between this amino acid and that amino acid. An attraction between this one and that one. These two are also attracted to each other, but there's no hydrogen bond between them. The attraction here could be simply van der Waals forces, and that is two nonpolar amino acids like to be around another nonpolar amino acid. So they are attracted together. Nonpolar amino acids would be attracted by a very weak force. It's called van der Waals forces. You don't really need to know about it, but if you think about it, when you have your oil molecules all together in the bottle of oil, why is it a liquid? It's because the oil molecules are nonpolar. They like being around other nonpolar molecules. But why is it a liquid? It's because the van der Waals forces are holding the oil molecules together in a liquid. And if you were to put like the olive oil in the refrigerator, the olive oil will no longer be a liquid, at least part of it would become a solid. And that's because the cooled temperature of the refrigerator has broken the van der Waals, well, prevents the van der Waals forces from breaking. And so that weak force is strong enough to lock the olive oil together, and then you have solid olive oil. Then again, if you were to break all of the van der Waals forces by heating the oil up, you would make the oil gaseous, and then the van der Waals forces will not hold the oil molecules together, and then you don't have liquid olive oil. Any question about any of that? So van der Waals forces, all you need to know are when two nonpolar molecules like to be together, like two amino acids, which are nonpolar. Now, these amino acids are being repulsed from each other, and that could be a nonpolar amino acid and a polar amino acid. They don't like being together. Another one could be if they both have a positive charge, like that. I think arginine has a positive charge, and I don't remember what amino acid and another one has a positive charge. They would be repulsed from each other. Two amino acids, like maybe this one here, although that sort of looks like it has a hydrogen bond, is attracted to each other. They can also have one that has a positive charge and another amino acid that has a negative charge. They would actually have an electrostatic attraction between the positive and the negative charge. And so the secondary structure comes about by many different attractions. Hydrogen bond is a main one, but there are other attractions that can come together, like the positive and the negative attraction, the van der Waals forces being attracted together. And those are three different forces of attraction that can bring the molecule together. And then there are forces of repulsion that can bring the secondary structure into function or into shape, and that is the repulsion between a polar and a nonpolar amino acid. The repulsion, this would be a good one here. This is the same one. If that has a positive charge and that has a positive charge, they would be repulsed from each other. Any question about any of that? All right. The video did talk about you can have the alpha helix shown here. You can have the beta pleated sheet shown here. And I guess the beta pleated sheet, let me see how that goes. Oh, here it goes here, and then here, and then here. And it's being held together by hydrogen bonds. I think these are supposed to be hydrogen bonds right there. The third one, which he did not mention, let me draw that, is the random coil. I need to draw the random coil anyway, so let me draw that here. The random coil will be certain regions held together, different regions repulsed from each other. So this amino acid here is repulsed from that amino acid. That's why they're further apart. This amino acid here is being attracted to this amino acid. This one here being attracted to that one there. This one here attracted to this one here. And that one there being repulsed from this one here. The random coil just has certain parts of the amino acid being attracted together, other regions not. The random coil is closer to the third dimension of a protein structure than 2 dimensions, because the interactions here and here are not really localized. And then these aren't localized here. And this one is being repulsed from this one here too. So remember, the secondary structure is the localized attraction and repulsions between the nearby amino acids. If it is attractions and repulsions between amino acids which are not nearby, that is the tertiary structure. The tertiary structure gives the overall 3-dimensional structure of a polypeptide chain. This is the interaction between amino acids all over the protein. Like right here, let me blow this up. Here we have an amino acid near the end of the chain being attracted to an amino acid near the end of the chain versus near the beginning of the chain. And here we have an amino acid midway through the chain being attracted to amino acid midway through the chain. This is an ionic attraction where we have a positive charge and a negative amino acid being attracted to each other. Hydrophobic interaction between the amino acids being van der Waals forces. There is a hydrogen bond being attracted between this amino acid and that amino acid. And here we have a disulfide bridge, which is an interaction you do not see in the secondary level. You only see the disulfide bridge being an attraction in the tertiary structure. So the tertiary structure has all of the forces of attraction and repulsion of the secondary structure, as well as the tertiary structure has the disulfide bonds which form between the cysteine, 2 cysteine amino acids. So here's a cysteine here and a cysteine amino acid there. Let me show you that in my drawing here. Let's make another. Let me break this down. Something like that. Okay, we have a cysteine right here, which has an SH group. I don't know if I can draw an S and H. Not easily. Not bad. And then there's hydrogen here for another SH group. Oh, that's a pretty bad H there. Oh, that's not so bad. And then an S here. And here's the cysteine right here. Amino acid. The hydrogens come off of the sulfhydryl group. So let me erase the hydrogen. And then the sulfur groups form a chemical covalent bond between them. This is called the sulfur dibridge. What is that called exactly? Disulfide bond. So a bond between two sulfurs from the cysteine amino acids. This is a chemical covalent bond between two amino acids. It is much stronger than a hydrogen bond. And for an example of a hydrogen bond, my mother used to put her hair up. The internet connection is breaking again. So if you didn't hear that, if you're not hearing this, let me repeat it. My mother used to put her hair in a curling, what's it called? The curling, I don't know, curling device. I don't remember what it was called. And she would put her hair in curls and put the I don't know what it is, the curl in her hair, and then wrap the hair around the curl, put a bobby pin to hold it in place, and then put a bunch of those in her hair, and then put the curling hat on and turn on the machine. I think it would put steam or warm up her hair, and she would get hydrogen bonds to form in her hair, and then she would take the hat off and take the curl out and the bobby pin out, and she would have her hair in this curl. And so she would curl her hair. And the trouble was she was living in Western Oregon, and so she would go out into the Western Oregon mist or high humidity, and the hydrogen from the air would get in her hydrogen bond and break the hydrogen bond, and her curl would sag. There. That was curling your hair simply using hydrogen bonds. On the other hand, my mother never did this, but you could go in and get your hair permed, in which case you have these links or kinks in your hair, and that forms the sulfur dibridge between the cysteines and the hair. And if you don't know, you can curl your hair and you can step into the shower and wash your hair for about a month before your perm will go out of your hair. After about a month of washing your hair, the perm will slowly start to fade. So that's how strong the disulfide bridge is in the perm, and it's because the chemical covalent bond is a very strong bond, much stronger than the hydrogen bonds. Anyways, in the tertiary level, we have all of the interactions we saw in the secondary level, as well as the disulfide bond, but these interactions can be among any amino acid in the protein. And the secondary interactions are only between neighboring or nearby amino acids. Any question between the secondary and the tertiary level of protein structure? Now, some proteins only can go up to the tertiary level. They do not have a quaternary level. These proteins are proteins that only have one polypeptide subunit making the protein. Every enzyme I know of only has one polypeptide making up the protein. And that would be something like catalase, polymerase. There's only one polypeptide subunit making up the protein. But if there's more than one polypeptide subunit making up the protein, these polypeptide subunits have to come together and then interact, where you have the same amino acid interactions, attractions, and repulsions that we found in the tertiary level, now in the quaternary level, and then the polypeptide subunits come together to make the functional protein. Like hemoglobin and antibodies have 4 polypeptide subunits that have to come together to make the final quaternary structure of the hemoglobin and the antibody. Any question about any of that? All right, so those are the 4 levels of protein structure. You need to know that proteins have a shape. There are globular proteins. These are roughly compact spherical proteins, like hemoglobin is a globular protein. And then there are fibrous proteins, proteins which are thread-like, collagen. The other structural proteins tend to be fibrous proteins. There are conjugated proteins. These are proteins which are a protein connected with another organic or inorganic component. The conjugated protein is named by their non-protein component. Like a glycoprotein is a protein hooked to a glycogen, and that would be a carbohydrate. A glycogen is a carbohydrate. So a glycoprotein is a protein connected to a carbohydrate. A lipoprotein is a protein connected to a lipid. So proteins are more complex and diverse than the other 3 biological molecules, meaning they're more complex than lipids, carbohydrates, and nucleic acids. Proteins, when they have their normal shape, they'll have their function, but the protein can come out of its normal shape, and that we call protein denaturation, where the protein loses its 3-dimensional shape, breaking hydrogen bonds, breaking other attractions, forming hydrogen bonds when they shouldn't be there. That can lose the protein's 3-dimensional shape. An example of a protein that will form a hydrogen bond when it shouldn't have a hydrogen bond would be one of our enzymes. If you were to put it in the refrigerator, the protein will be more compact. It'll form hydrogen bonds that it shouldn't have. And like I said, you can also heat up our enzyme, break the hydrogen bonds, but you can break other bonds also to lose the protein's 3-dimensional shape, which will begin protein denaturation. If the protein has no longer any function, the protein is totally denatured. The protein has totally lost its shape. So when you're heating up an enzyme, the protein will start to lose its shape, the protein will start to denature, and then let's say you boil the protein, you totally change the shape of the protein is totally denatured. Proteins can denature if they're put in the incorrect temperature, the incorrect pH, the incorrect salt concentration. Any question about any of that? All right. Any questions about proteins? If not, let's move on to nucleic acids. There are 2 nucleic acids, DNA and RNA. DNA, the long name is deoxyribonucleic acid. RNA, the long name is ribonucleic acid. Nucleic acids consist of a monomer called a subunit, or the monomer is called the nucleotide. Nucleotides themselves consist of 3 parts: a 5-carbon pentose sugar, which for DNA is always deoxyribose, And for RNA, it's always ribose. Nucleotides also have a phosphate group shown here. That would be the 5-carbon pentose sugar. And then they always have a nitrogen-containing base shown here. Any question about any of that? There's another nitrogen-containing base. There are 4 nitrogen-containing bases in each of the DNA or the RNA. There are 2 purines and 2 pyrimidines. For DNA, the purines are adenine or A, guanine or G. The pyrimidines are cytosine or C, or thymine or T. How to memorize the purines for the pyrimidines is the word pyrimidine has a Y in it. Cytosine and thymine each have a Y in the name. Adenine and guanine, the purines, do not have a Y in the names. And that works for DNA. Now for RNA, the purines are adenine and guanine. The pyrimidines are cytosine, but RNA does not have thymine. Thymine is replaced by uracil. And you simply need to know that thymine is a pyrimidine and uracil replaces thymine. Uracil does not have a Y in the name, but it replaces thymine. Thymine has a Y in the name, so uracil and thymine are both pyrimidines. DNA and RNA can bind to each other. RNA in our cells doesn't tend to bind to each other, but DNA does. And the DNA always binds to its complementary base. A and T always bind together, or T and A always bind together, and C and G always bind together. It's the hydrogen bonds between the A and the T and the C and the G, or the vice versa, the G and the C, which form the bonding between the complementary bases. A and the T always have 2 hydrogen bonds between the A and the T. C and the G always have 3 hydrogen bonds between them. And I kind of have it shown here. It's not the best, but it's the only one I have. The C and the G have 3 hydrogen bonds between them. Any question about any of that? All right. DNA is composed of the sugar deoxyribose. DNA is normally double-stranded in our cells. There are exceptions where the DNA is not double-stranded, like whenever the DNA is being replicated, the portion that is being replicated is only single-stranded. But as soon as the DNA is replicated, it'll become double-stranded again. There is another exception, and that is when the DNA is being transcribed, the DNA will be single-stranded. But usually in our cells, the DNA is double-stranded. The DNA is held together by the sugar phosphate backbone. Let me see if I can blow this up. So here we have I don't know, it's not shown, but sugar, phosphate. The nucleotide is on the side of the sugar, I think, and it is not holding the DNA single strand together. It'll be the phosphate, sugar, phosphate, sugar, phosphate, sugar, holding the single strand of DNA together. And that's a covalent chemical bond holding the single strand together. It is between the nucleotide, which complement each other, meaning the G and the C complement each other, and the A and the T complement each other. It's between the complementary bases that the hydrogen bonds form between the 2 strands of DNA. And the 2 strands of DNA are only held together by the hydrogen bonds between the complementary bases. Remember, hydrogen bonds are very weak. And for example, in this hydrogen bond right here, that hydrogen bond will break and reform all the time. But at least at our body temperature, 37 degrees, this hydrogen bond will break and reform all the time. When this hydrogen bond breaks, this one and that one are holding the DNA together. And then this one will reform and that one over here will break. Any question about any of that? So it's the hydrogen bonds which hold the double strand or the two strands of DNA together. And the hydrogen bonds are only between the complementary bases on each strand of the DNA. DNA is also twisted in a spiral staircase, like a spiral ladder or a staircase. And the hydrogen bonds and the the nitrogen bases make the steps of this ladder. They are the steps. They are not holding the single strand of DNA together. Any question about any of that? All right. RNA is composed of the sugar ribose. RNA is normally single-stranded. The sugar phosphate backbone holds the RNA together. Once again, my internet connection is unstable. So the sugar phosphate backbone, once again, holds the RNA molecule together. The RNA is normally single-stranded. The bases are A, G, C, and U. The T is replaced by the U. There are three major kinds of RNA. In cells, tRNA or transfer RNA, mRNA or messenger RNA, rRNA, and or ribosomal RNA. All these RNA molecules are the same. The only difference between them are the sequence of the nucleotides. And so all you can tell them apart is simply sequence the RNA, and if it follows the messenger RNA sequence, it's a messenger RNA. If it follows the tRNA sequence, it's a tRNA. And if it follows the rRNA sequence, it's an rRNA. The RNA genes are encoded by an RNA gene. Most of the genes are coding for messenger RNA, but there are about a dozen RNA which codes for tRNA and a dozen RNA gene coding, actually less than a dozen RNA genes coding for rRNA. Any questions about any of that? All right, if not, I'm going to stop here and I will see you at 6:30 for the lab. Any questions?
Will this be uploaded after we finish it?
We're not finished with it, so it will not
Yeah, okay.
be uploaded until we finish it. All right. Any other questions? All right, bye.